Pennsylvania. He's a serial entrepreneur with over 35 years in the biotechnology pharmaceutical industry. He's he has been 30 of these 35 years has been based in North America in both corporate and consulting capacities. He consulted a variety of life sciences topics with particular interest in bioprocess development, manufacturing, in addition to application of quality compliance principles to modern processes. He has acted as an expert consulting for the FDA on CGMP and data integrity issues and assessed both domestic and foreign companies with quality compliance. As part of his government service, Mr. Nigel has assisted both BARDA and the Department of Defense in the relation of production of anti-terrorist countermeasures. He has a special interest in the application of lean process excellence principles to bioprocess manufacturing systems, and he's the author of a book dedicated to this topic entitled Lean Biomanufacturing by Oxford Health Publishers. We welcome you, Mr. Nigel. We're very proud to have you here today with us with a fantastic and very, very new topic to the region. I'll leave everything to you now. Now you can take over and you can start your presentation. Thank you, Mr. Nigel. Go ahead.
Yara, thank you very much for that very sweet introduction. It's very, very nice to be with you all this evening in your region, and I'm hoping that some of the things that we can share with you today will add some value to your own circumstances, and hopefully it'll be able to make a difference to the way your companies operate. Certainly I hope that's the case. What I want to do first of all is to start with a very clear understanding of how these lean principles work. So I want to run through some of that with you. I think that's really important because it's going to set a foundation for explaining some of the things that we've done with companies that really have made a tremendous impact on how efficient they run in terms of cycle time reduction, increase in throughput of samples through their area. But not only that, it's the integration of the laboratory properly with the manufacturing side, and then also with the quality assurance release of products. And I firmly believe that if you do not have all those aspects coupled together properly, then you may have a very efficient lab, you may have a very efficient manufacturing end of things, but you're going to have these big wait times in between where there's no value addedness and where time is lost, and so money is lost. So from these principles, we can better design our lean lab area and make it more workable from an efficiency point of view. It'll also have a dramatic effect on the quality compliance of how you process your samples in your laboratory area. You will notice that as you apply these techniques and tools, then the number of mistakes, the deviations, the compliance issues will drop right first time as a principle will increase. All those issues will have a dramatic effect on the throughput of samples because you're not going to be doing as many investigations. And all that will have an impact on how quickly you can release your product. And I think really, so that's going to be very important. So we're going to transform from what we're going to call the current state, from the way you're operating now, to a future state or an ideal state. And those are the things that are going to be set as our objectives for the talk. And I will go through that. And I will go through that quite quickly, make notes, and I'll answer questions towards the end. The last 10 or 15 slides will have pictures, examples drawn from one or two clients that have given me the okay to show you those things. Obviously they're anonymous, but you can see the types of improvements that the ideas here will make. Okay, so let's go shoot. So we're going to leverage the lean principles, the process excellence principles, to hopefully generate a holistic and sustainable approach. The whole idea behind lean as a concept is to use less of everything to produce more. So we want to use less time, less equipment to actually turn around more samples in the case of the laboratory. So that's basically the foundational. We want to generate also an operational culture that removes the waste out of everything that we do. Waste is not just paper bags or plastic bags or used pipette tips. It's everything within the supply chain, both in the manufacturing area and the lab area, and then later in the distribution and in the quality end of things. What we're trying to emphasize here, although we're focusing on the lab today, we're focusing on, although we're focusing on the laboratory today, we're actually stressing the importance of the lab as an integrated part of the whole enterprise. And so you need to be thinking about that as you develop your lean principles for your laboratory. So we're looking for a continuous improvement environment that's directly associated with this whole enterprise, not just the lab. So think outside of the laboratory for part of the solutions to what's going in your laboratory. And if you do that, then that will create an environment and a culture that isn't we're the lab guys and you're the manufacturing guys. If you your facilities are, I'm sure, very similar to those around the globe, and there's often complaints that the manufacturing people don't send the samples to the lab people soon enough, and the lab people are always slow for the manufacturing people. So what we're trying to do is to create an environment whereby, are you ready? And do you have the mindset to operate in this mode? That's a very, very important part of embarking on one of these lean lab transformations. So some things that we need to focus on: control of our raw materials. Yes, we've got lots of things that are going to be associated with the lab, lots of consumables, lots of components, lots of reagents. We need to make sure that all those are lined up properly so that we're not causing delays. I'm going to share some concepts with you in a minute and some terminologies. You need to really make focus on those because it's going to impact everything that we do going forward. So we need to effectively control the processing time. If we think about high pressure liquid chromatography for a particular product, that's going to be a certain set time. Yes, that's true. But we don't want things to be sitting around wasting time trying to get equilibriums with our buffer systems. System suitability, we need to have that nailed down so that we can take advantage of the other things that we're doing. And then we need to make sure that we've got tight control of our equipment. We need to make sure that the columns are properly set up. We need to make sure that the things are properly calibrated so we're not wasting time. If we're dealing in a multi-product plant, and I suspect some of you are operating multi-product plants, then we need to have really the changeover mechanism from where we're exchanging columns, where we're exchanging elution buffers and things like that. We need to have that nailed down so that we can change over from one set of equipment requirements and specifications to another. And then allied to that, we need to make sure that we're using space properly. I have a slide towards the end, which I'm going to show you, which is a spaghetti diagram. A spaghetti diagram is one of these red line diagrams that basically shows you the flow of personnel or materials or product or samples that go around a typical laboratory. And you can see the flow patterns are horrendous. They're very poor. They're very inefficient. And they are causative and lead to mistakes. And the mistakes are the things that we're trying to cut out because the mistakes cause non-value added work to be done, and that's both time consuming and costly. So we need to make sure that we've got the specification of the best equipment to do the job. Whether it's, are we using a UPLC in place of a HPLC? Are we using a chromatograph, gas chromatograph setup? Are we using, the right equipment for the right test system that we're doing, that we require? We need to set those things up. What we're trying to do here, what's very important, is we're trying to sequence the activities so we get what we call smooth flow. I'm going to show you a diagram a little bit later on that shows you what a typical laboratory looks like in terms of going from a lot of work all at once to very, very little work to lots of work. And it's basically, you end up with a sine wave, which is very inefficient, where you have too much work for the resources, then not enough work for the resources. What we're trying to do is to smooth that out so that we have a constant supply of samples coming in, and we arrange the staffing and the equipment use to maximize the uptime of the equipment, but also the uptime and focus time of the analysts. The analysts are highly trained, quite expensive. If we can substitute some of the things that they're doing for technical people that are assistants, we want to do that so that we can maximize the attention and skill of the chemist or the pharmacist that's doing the analysis. We need to make sure that we mistake-proof equipment and processes. I can't tell you how many times that we see in laboratories and support areas where operations are lost because there's the opportunity to throw something away, or they'll throw a wrong valve. If you can mistake-proof things that will not allow you to throw away your sample, then you're going to be in a much, much better situation than if you don't do that. It requires continuous attention to making sure that your equipment is properly calibrated. If you don't have that equipment calibrated properly, then you will lead to more problems. So the underpinning philosophy here is waste elimination. So what do we really mean by that? So let's look at what we're talking about. Lean comes from Japan as a concept, Lean Six Sigma. MUDA is the term used to describe generally all waste systems, and it's commonly recognized that there are 7 forms of waste identified that can be applied to your systems. So let's look at those. There's an acronym here that we call TIMWOOD, which categorizes the 7 forms of waste. Transport waste is associated with the data and things, so anything that's requiring movement is associated with transport waste. Inventory waste is associated with poorly managed stock or other raw materials, for example. Anything that is associated in this case with doing your analysis. You're probably asking, well, what's this got relevance to the lab? We'll show you when we get to the configurations for a modular arrangement why this is so much more preferable than having things spaced around the lab area, as you often find in a traditionally operated laboratory situation. Motion waste is associated with the analysis and elimination of unnecessary personnel movement. If you've got, for example, materials, glassware that's at one end of the lab and solvents at the other end of the lab and something else, and you have in the center of your lab your weighing systems, and then in another corner your analysis equipment, then you can see that the movement of both the sample and materials and your analyst is walking backwards and forwards all day long. This is very inefficient. It also leads to problems if you have a rather crowded lab, which when you get busy you will, and people bump into each other, or people put things down, or equipment is left not properly cleaned. So layout and structure is very, very important. Waiting waste is caused by delays and backlogs. What we're trying to do with smooth flow is to avoid backlogs. When you get backlogs, people get angry in, for example, if you have a backlog for your in-process samples. That's not good, because if they're waiting for an analysis to actually do the next reaction in a multi-stage chemical synthesis for a drug, then that means your plant is idle until the analysis comes back. So you want to be able to phase, you want to be able to forecast when that sample's going to arrive, get it logged in, process it quickly, do the analysis and get the result back. It may be, for example, with one of our clients, if that wasn't got back within 2 hours, then the material that's now an intermediate could actually go off. And so you would be into a rework situation. So a rework is not good. It's more money, it's more time, and it affects potentially the number of batches that you can get out of a particular manufacturing train in a month or in a year, for example. So waiting time is very, very important. It causes delays and backlogs. When you get backlogs, the human condition is such that it requires you to speed up. And when you speed up, you make mistakes. So overproduction associated with making too much. This could be if we generate too many samples, again, for the demand potential of the laboratory. And overprocessing is associated with unnecessary handling. With a lot of laboratories that haven't moved to a lean environment yet, you'll see that the sample is being handled as a chain of custody issue, where the sample comes in, it's maybe logged into a manual system, it then maybe goes into some sort of bin arrangement or a retention area, somebody else picks it up, and there are too many potential sets of hands, which can cause a problem with mistakes that get made. Again, the mistakes is waste, and so that causes us time. So what we're trying to do in terms of our layout, in terms of the technical operations that go all the way back to login of the sample, to apportioning the sample to the right analyst, to actually performing the tests, making sure that you have the right test analysis sheets, right through to doing the analysis, making sure the data is logged in somewhere, whether it's a LIMS or some other data device. And then obviously the data has to be approved by maybe the head QC chemist before it's then handed on to quality assurance for release. So defects could be any defect. In our case, for the lab, we're talking about the analysis could be an OOS. So we want to minimize the possibility for OOSs. So there we are, Tim Wood. So waste elimination is foundational to everything that we do, because it really embodies the way forward to smooth flow. And with smooth flow, we get better analysis, shorter sample times, cycle times, we get fewer mistakes, we get more right first time, and everybody as a result of that is pretty happy. So remember, less waste means less work in process, lower operating overhead, shorter cycle times, faster changeovers, and reduce lead times. If you know a sample's coming, then and it's coming at a certain time of the day because the sample's going to be pulled, it means now that you can again set up your, if we're talking about HPLC, we can set up the system suitability to be so that the machine's ready and stable when your sample comes in. You allow a certain amount of time for workup and prep of the sample, and then you inject or you put it on a carousel, and the thing goes through maybe it's 25-minute or 45-minute sample cycle until you get your analysis, and so that's a critical component. So tools in our lean toolbox that we're looking at, waste removal, we've gone through that. Process streamlining, cycle smoothing, cycle time reduction, mistake proofing. They use a word called pokiyoki in Japan. All it just means is mistake proofing, and then rapid changeover. They have another word there which I didn't put in, which is called SMED. You don't need to know that. As long as you keep these core principles in place, then that's really what you need to know. Okay, we just mentioned those. Value stream mapping is going to be very, very important. I'm sure many of you are familiar with process flow charting. Value stream mapping is a little bit more involved, and now I'm going to explain what the value of that is to you. It helps you to focus and interpret really what's going on. It also really points you towards what you need to do next with your business, in this case the laboratory, but it could simply apply to, again, the manufacturing area, or the integration of the manufacturing with the lab, and then the lab with the quality assurance department, to make sure that that there is a like a baton handoff. If you think about athletes running 4x400 meters in a relay race, you hand the baton on from one to another, and eventually you win the race. If you approach this in the same way, what we're trying to do is to hand the baton on rather than having to stop, pass the baton on. It's going to mean that you will probably knock 10 seconds, for example, off your 4x400 meter race, compared to if you hand it off within that little window where you don't stop sprinting. So that's the idea, what we're trying to do here. We're going to apply the principles of Professor Deeming back from the 1950s, and more recently from the microelectronics industry, using a DMAIC or a plan, do, act, check methodology. This is the basis of Kaizen. This is the basis of how we do our transformation from the current state, which is where you are right now, to hopefully implementing a lean lab strategy within your whole manufacturing enterprise. So what we're doing here is we're going to identify the opportunity for improvement. So in this case, we're going to say, we've got HPLCs at one end of the lab, we've got glassware at the other end of the lab, we've got a washing area, we've got a prep area in another corner. So that is, in this case, could be the problem. We might want to identify a root cause. Well, the root cause here is quite simply there is no planning. So we define what the future state looks like. We want something that's compact, where it could be all together. That would be the ideal solution. So then we say, okay, let's brainstorm. We call it try-storming here in the US. We try things out. When we brainstorm ideas, we come up with maybe 3 or 4 ideas, and we try them out sort of hypothetically. We may create a model. We may, in some cases, with one of our clients, we created on a separate floor a test bed area to see whether or not it would work in their environment. My team knew that it would, because it's not the first time that we've done this, but clients need to be convinced that it works before they will reorganize their whole laboratory situation. So that's what we do. So you try storm those ideas, and you come up with something that actually looks better. What you need to do then you need to clearly understand what it is you're trying to do, and you need to know what is it that bothers your customers. Your customers are the people, in this case, if we think about in-process samples. Your customer could be the manufacturing group, because you want to get the customer their sample back for their analysis, so they can continue on with their next reaction. So what's bothering them is, in this case, for example, would be that it's not getting back quick enough. So we're going to look at what do we need to do to the lab to make it more flexible, so that we can achieve that as an endpoint. If we look at our situation, what's causing us money and resources? What aspect of whatever we're doing fails? Is it the way that we're making up the system suitability buffer? Do we have everybody making up their own buffers in the laboratory? Do we have a technician making up buffers for the day? what things are going to impact the probability of you getting your analysis back quicker and with fewer mistakes. So it's really important to focus on the process and focus on what solutions rather than on people. Unfortunately, life being what it is, we tend to there tends to be a lot of personality conflicts in companies. This is why it's very important to establish a collaborative team approach and show before you ask somebody to do something which is going to cause them some work or change something which is different from what they've done before, if you can persuade them and show them that actually it's going to benefit them, then the likelihood is that they're going to join with you and be a lot more cooperative. And cooperation in a lean environment is very much the name of the game. So when we look for our solutions, when we look at the problems we have a phrase that we say, don't be too broad in terms of the things that you're trying to fix. Look at creating narrow improvements, but make sure that they're far-reaching. So be very focused, an inch wide, go a mile deep. Really understand what the implications are, because that way you have a greater probability of success the first time around. One of the things about the PCDA cycle is you don't just do this once, you do this many times. Even when you've implemented your lean lab, what I tell clients is this needs to be part of your operational lifestyle. It's like if you want to get fit, you don't go to the gym once, you build it in as part of your lifestyle. It's the same with these process improvement programs, that you must exercise your efficiency, you must exercise your creativity constantly, because you can always improve. We're working in a very dynamic environment, and so there's always change going on, and if there's change going on, then you have the opportunity to make an improvement. So if you can connect, and I've mentioned this, we're looking at the lab, but if you can connect what you're doing with the manufacturing, and then at the other end, the quality assurance, that's where you're going to see a big benefit. And if you can co-opt, if you can invite the members of the manufacturing to supply your samples at a certain time, and you can persuade the quality assurance people to receive your analysis data at a certain time, then the overall cycle time for the factory is going to be reduced. And you're in a situation where you win. You create a winning environment, not just for the lab, but you create the winning environment for the whole process. I have a saying that everybody likes to be a winner. So basically, you need to share in all the accolades that go with creating a winning environment. So focus on the flow of the sample, the product, the information, not the problem or the defect. Remember, these are the results of poor flows. To be successful, you need, as I say, a deep understanding. Look at all the activities involved. Look at the resources that are in play and where they've come from. The flow of people in the lab space, the material flow, the raw material flows, the in-process samples, they can all be different. You need to look at those. As I say, I'll show you a spaghetti diagram. You need to do spaghetti diagrams to elucidate how that is working. It's good to draw pictures. It's good to take with the smartphones today. It's very good to do little video clips to see how things are done. Just as we find with our procedures that something is supposed to be done a certain way, when we actually come to see very often how something's done, it's not quite the same. And where you have a lot of people doing the same thing, Everybody's doing it the same way, but slightly different. Videoing those situations is a good way to kind of freeze that, so that you can sit down as a group and say, hey, this is what's going on. Do we all see what the problem is here? And then you would design a Kaizen, which is the plan, do, act, check. You would do a Kaizen event, and you would come up with a series of things that you want to change and objectives that you want to achieve, and then you would perform the Kaizen. Okay, I think we've covered that. Current state. Take the photo of the current state. Use any data available to help gather the details versus what is, and as opposed to what it should be like. Very important. I threw this one in late yesterday. That's why it's a slightly different color. Scorecards are things. I think it was Einstein that said, you know, doing science without good measurement is madness, or something to that effect. Making changes unless we can measure how well we've done is a waste of time. So we need to try to generate metrics for an improvement, and to generate those metrics, we need a scorecard, which is some sort of a deliverable which will allow us to assess the performance of what we've just done. We want to use like a standardized format and language to avoid it being ambiguous. We need to be specific about things in terms of driving the performance opportunities. It's also good to show management what we're doing, so that's a useful tool. That communication up the ladder is always good. It's also a good tool because it empowers the workforce at the grassroots level. I want to say that it's very, very important that the grassroots are empowered to do this, not for the management driving it. The grassroots analysts in their laboratory space need to power this with the support of their management team, but let the operators do the Kaizens, let them suggest what changes are made. They know what are things that make their work environment non-productive. We've done some very, very good exercises with people that we've just said, make the change yourself. And they look at you to say, what do you mean? Because they're not used to making changes. When they make the changes, they own it. When they own it, they perform that way every time. And when they see the results, that gives them a winning attitude, and they want to make the next change. That's the way you get sustainable performance, and that's the way you get continuous improvement. So you can, when you do these Kaizens, you can write down a checklist, and this is just an example of what we're talking about. You want to know what the conditions are. You want to know who's, you know, who was making the decision, what materials, how are we going to measure performance, what the quality is. So you can just generate that. This is not a static chart. This is something that is just an example. I mentioned flow charting, and the real dynamic tool here now is value stream mapping. Value stream mapping really gets to the heart of the exercises that are involved. It's basically where you chart out where the non-value added and value added in any operation that you're performing is. So you can see here that it's specifying an activity. That activity could be a login, for example, and it takes a certain amount of time. Then there's a wait time where the sample sits around with a log sheet. Then the activity could be prep time, and whatever amount of time that takes. And then there's a wait time because the sample can't go on the machine straight away. The next activity could be the analysis, and then the next activity could be the the data analysis. Some of these will be really added value, and some won't. Added value is created, is usually termed whatever the customer will pay for, and it's a rather strange way of describing things, but it's you only want to be doing something if it adds value to the product, then that's value added. If it's not, then it's not something that if I was buying this, I would want to pay for. That's just your inefficient operating situation. You know, I'll pay for the analysis, but I don't want to pay for all the inactivity associated with the fact that you can't run your place well. So then what we do in this, we circle the activities that are providing value. Clearly, the login, maybe not value added. The prep time adds a certain amount of value, and the analysis time maybe adds value. There are maybe in any area there are going to be several of these. We've done analysis of complete processes with companies to find out how we can reduce these wait times, which is waste. And we've established that earlier on. So the idea of what the current state looks like will be generated once we've done this value stream mapping, and it will give us a basis of how we want to design the future state. The future state will be arrived at by now focusing using that PDCA and doing the Kaizen event to actually correct each one of those facets that's not functioning as we want. So in this case, we might record the cycle time. We compare the takt time. The takt time is a cadence. It's the drum beat. It's the you are taking a certain amount of analysis, and it needs to be performing at a certain drum beat rate, at a certain cadence, like on a bicycle, to make the difference. To make a difference. And the takt time that we may be running at might be 2 hours, 2.5 hours, and we need the cadence to give a takt time of 2 hours, because we need 2 hours to turn around the analysis. So we have to up our game, and so how can we do that? We do that by looking at the value stream mapping, and we see where the holdups are, and we make corrections by doing the Kaizen activities within those. So here we go. We develop the individual Kaizen improvement projects to adjust the areas that require improvements. We make the changes. We develop and see if we meet the takt time. How do we know that? We've set up a scorecard to measure that, and then we repeat the process if it doesn't work. The scorecards will make sure that the changes being made are appropriate, and we do this by setting the metrics in place, and then we track those metrics and we trend them. Remember, I've said this is continuous improvement. Once you've got something where you've said, hey, we're running at 25% improvement, then why not ask the question, why can't we run at 30% improvement? Well, what would we need to do that? You'd look again at the value stream map. You would look and see where the deficiencies are. You would do another Kaizen. You would set the events, and then you would perform that. You would measure it and see, are you meeting your targets? And you would do that over and over again. And you would track that improvement. And over the course of, say, a year, you can then do a kind of a process capability plot, which will give you the opportunity to see if the investment that you've made in moving to a lean lab is actually profiting your organization. As I say, you can do the key performance indicators daily, weekly, monthly, or annually. Most of our clients would look at them on a weekly basis, and then they would present this data in a series of charts that they would post in the laboratory or just outside the laboratory. So the analysts, as well as management walking through the facility, can actually see how well you're doing. That's a really important part of this exercise, communication. You need to communicate to people how well they're doing. It's no good just chewing people out when they're not doing a good job. You need to reinforce positive attitudes, and the best way to do that is to declare the results that you're getting and celebrate as a group when you have exceeded expectations. It's important to note that QC labs are often isolated from manufacturing areas. They've certainly, in older facilities, got the short end of things. They usually get stuck wherever the space, and that creates its problems. So we need to organize ourselves in that space. If you have the opportunity of a new facility, then that's great. We can get things right first time. But if we have to reorganize, then doing so along the lines that I'm going to show you now is going to be vital for you to do that. Again, try to see the lab as part of the greater enterprise. There's a term here called Kanbans. It's another Japanese term. All that means is that as we move to smooth flow, a signal at the end of analysis, at the end of the analysis, is signaling that that there is a need for the next set of samples to be brought into the analysis stream, so that we don't create peaks and troughs in our analysis cycle timing. So key points, again, oscillations from the operating norm cause inefficiencies. Balanced flow paves the way for better use of resources. It prevents backlogs, and the consistent utilization of equipment, instruments, and people is more efficient, and the reliability of the analysis goes up. The right first time is a bonus. It's fair to say that UQC labs, most of the ones that we've worked in, are very highly stressed environments. So we need to make sure that we build in and keep things that prevent us to have work in progress. WIP is a big inefficiency in a laboratory, as it is in a manufacturing plant. So by making sure that analyses of a particular sample that might have 4 or 5 analyses required of it before a decision point can be made, it's important that those are properly tracked, logged in, and prioritized. We can flow that one. So the volatile nature and workload of the samples can stress the equipment. So we're looking at a more controlled, smooth flow. So what creates these instabilities? Those of you that operate labs know that you'll get things like late stability sample pulls. They're I wouldn't say rarely on time, but they're off time, plus or minus a day or two sometimes. You'll get in-process samples that maybe come off late. Some of the things I'm going to show you with one of our manufacturing clients, we've been able to ameliorate that situation. Validation samples that are coming through for a new process. If you're making a new product coming through, that can generate a lot of extra samples when you've already got a normalized workflow. That's going to cause a perturbation in the operation, and so you need to be well set up with this kind of a lean approach to accommodate that, so that it doesn't cause instability. A new product introduction, as I say, is an example of that. Sometimes at the end of a shift, you'll get a priority request. One of the things that we try to discourage is special cases. We all do it. We see it in every company. It's important not to have one-off special cases, because what happens is that gets out around the factory, and then somebody knows somebody, and they'll put in a special request for an analysis to be done, and then it gets to be an organizational management issue, because every sample now becomes that #1 priority, and that just can't be. So this is what I've been describing verbally so far. The Heijunka tool is a way that we try and smooth flow. So the top diagram is showing you what the typical lab operates like. It's high demand at the top, then there's a trough, then there's high demand, and it's peaks and troughs, peaks and troughs. What we're trying to trend towards is a flattened out curve, a bit like what we've been trying to do with the coronavirus, which is a smooth flow. Okay. The samples arrive usually in a central area where they're labeled, numbered into trays, and arranged with various documentation. Using a visual factory approach, which we call visual factory, something that's highly visible, we can generate better standardized work patterns. We're very much in favor of barcoding everything, and I'm going to show you something in the last few slides which shows that. If you can barcode straight into a LIMS system or some of the data system, and then put these into trays that are also barcoded, a little bit like location, in a warehouse type of situation, then you can move things much more efficiently. The important thing is that we need to have a constant delivery of incoming samples, not 25,000 at once and then another 25,000 another 3 hours later. We need to keep the capacity fully utilized at all times. That's impossible, but in a multi-plant situation, by staggering the way the samples come from the manufacturing floor, especially if they're in process samples, that can be achieved. So what we're looking for is this kind of a clockwork repeating basis. So we can use something which we can call a carousel or a rhythm wheel. It's basically defining a known work sample, and using that known work sample, we can level out the delivery and expectation. We can level out the analysis and the delivery expectations to the customer. So this again is very stylized. This is no one in particular, but you can imagine on a 7-day week, you could do sterilities on one day. This would never happen. It would probably be different products that might be analyzed on different days or from different shifts. But you get the idea that it's a rotating mechanism whereby a certain amount of time could be 1 unit or 2 units on the scale that would be allotted to generate analysis of raw materials or sugar analysis or potency samples, for example. Having that consistent delivery of incoming material means that we know now, because we know what the analysis time, this is where you need to build standards for analysis and analysis times for doing the various analyses. You can calculate and program how many samples and what workload of samples and how many staff you need to operate a given set of requirements from your manufacturing floor. Yeah. There's another term here, creating pull. What we don't want to be in this situation is we want to be having, if you like, the supply end of the operation pulling the need to do analysis through. So whether it's raw materials, for example, because we know that we're going to be making a product next week, that will pull our requirement to do analysis of the raw material for, a bioburden or a nitrogen analysis or carbon analysis. So we want to set up a mechanism whereby we generate the minimum amount of lead time, so we have this just-in-time setup for getting analysis done, in this case for raw materials, so that it meets the requirements of the manufacturing floor. It reduces the lead time of the process. The pull created by the customer demand drives the release of the manufactured product. This, or it could release the raw material for use on the manufacturing floor in a different set of circumstances. This is enabling the design of the process to be ready, things to coincide as I said, so that you don't have a disconnect. If you have a disconnect, we're back into tin wood again. We're back into weighting waste, which is inefficient. So I did say that I was going to show you a layout that is very much something that we favor. So this is called a U-shaped workup area. You can see on the right-hand side, the sample comes in. There could be a lab notebook. It could be a notepad. It could be a computer station. And then you've got space. Everything that you need. This would be a bench. These could be cabinets underneath that have all the tools, all the drawers with all the spatulas, the glassware, the reagents that you need to do the workup of that sample. There could be a wet area on the left-hand side. Some of our clients have like a extraction device there that allows you to extract the material, and then it will be worked up on the bench in front to prepare the material for HPLC. Then, if we look in a line, you can see that these are modular units. So within a laboratory, you can set up a series of modular units where the sample flow comes in, and each one is generating material for a bank of HPLCs, which are also ganged up in line. This could be within the same lab area, usually separated by about 6 or 10 feet, or in some cases we find that the HPLCs are across a corridor, for example, in a unique room, and you can see the data as it's generated is piped off to a LIMS system or some other data analysis unit. So that figure demonstrates a U-shaped configuration, and for rapid HPLC or UPLC type of operations. The reduction in sample movement, looking at the benefits here. The benefits are reduction in movement, speeds up the processing time, gets rid of the non-value added. The analysis is not moving around as much, they're not bumping into people. And we've standardized the practice to such an extent now that it's very, very difficult for one analyst, and one analyst might be working in one of these units or in several of these units, but it's very, very difficult of them to make the differences in the way the workup's done, provided you operate in this type of environment. There's just a simplification of the process that allows and permits a greater degree of smooth flow, because you're working up all the samples at the same time in sequence, which then means that there's a delivery to the instruments for the final analysis. The level flow, these are some statistics for you. Better use of resources here, 50 to 60% in some cases, and even higher with one of our clients. The volatility reduction frees up time on equipment that was not previously available. So you can actually see an OEE, or a capacity utilization, increase in that space. The error reduction falls. You prevent a backlog, which is a major factor in speeding up process release. And as I said, the backlogs, if you run into backlogs, you will end up generating a lot of CAPAs, and CAPAs are expensive, they take time, and it's something that from a regulatory compliance perspective, the fewer CAPAs we can have, the happier and the faster you can get inspectors through your laboratory when they come for their GMP inspections. Again, fewer investigations means QA can function to work with an operation to get the products out on a regular cycling basis, rather than this week it's going to take, this week it's going to take 10 hours to release a product. Next week it might be 2 hours. Again, you're putting them into a similar situation where their daily routine is not cycling like a sine wave, but more like a flattened curve, like we've tried to do again with the virus that we're all battling right now. So in the future state, we need to design and focus on the placement of the instruments. the storage of chemicals, any electronic aids, and the interaction between the wet area and the analytical area. And I showed you the adjacent side-by-side, the adjacent side-by-side arrangement there, and that works very, very well and very efficiently. We need to make sure that the flow of the samples through the laboratory is managed, and we need to make sure that we keep the analysts doing what they're doing, which is working up samples, doing the analysis, and then maybe in some cases assisting with the interpretation of the data that's generated. We don't want them walking around the laboratory. That's inefficient, and it's not a good use of their skill sets. So as we get to the analysis end, here is a relationship diagram which shows you, as we're getting towards the end of the talk here, I've got a few more to show you with the diagrams. This is again the rhythm wheel with the QC analytical release process, which is showing you again how analysis can be done and the process, how we drive it, how it can be, we might need a lead time of 15 days, how we are analyzing several products at the same time, the role of management in an oversight way. We're trying to program, if this was a sterile product, for example, we might do all the potency testing and all those things early on in the 30-day cycle. But we would program the sterility test such that on day 29, for example, we get the analysis as the very last data point, which is entered into the LIMS system. And then at the end of that LIMS system, 30 days, basically the material can go off. All the data systems can go off to QA to release the product. And so there's no hold-up times. If there are problems, then we set up a T-card system, and then we have an offline team that investigates efficiently to do that. That can be set up as well electronically, which today would be more efficient. What I'd like to show you now is some practical examples of things that we've done which kind of help you quantify or crystallize for you the types of things that we've spent the last 45 minutes or so talking about. This is a smart board that we put in the QC analysis laboratory for a major client. This is also potentially duplicated on the manufacturing floor, where samples would be taken for in-process analysis and then sent to the QC laboratory. Let me explain what the problem statement was here, that the QC department never knew when manufacturing were going to send the sample through. Technically speaking, it was supposed to come within 2 hours. They would get a phone call, or they would get an email notice saying it's coming soon, and it wouldn't arrive. Or it would arrive and somebody working the night shift, for example, didn't pick up the email, or they would leave a telephone message because this was a rather big plant, a very big plant, in fact. And it was about a quarter of a mile away from the manufacturing floor to the QC lab. And so very often the QC didn't know that the sample had arrived, and so the analysis wasn't done. And so there was a lot of wait time, a lot of waste time. There was a lot of mix-ups. So what we did here was create a smart board which shows, and the color bars that you're seeing is when a sample arrives, it's scanned in using a barcode scanner, and then it's given a number. For each one of these is a particular sample. And for this particular analysis, when this was taken, the analysis time, the complete cycle time, was to be no more than 6 hours. So as the sample analysis started, it progresses in time from left to right. As you can see, the length of the bar is getting lengthened. If it should exceed 6 hours, then it would go to a notification in yellow, and if it went more than 7 hours, it would go red. And so what we created here was a very visual display whereby people in the lab always knew if something was a problem, running late, or if they had a delay, or if a sample wasn't analyzing well. But also the people in the plant could see at what point their analysis was being done. I'm happy to report that that cycle time started off at 6 hours and now is running around 3 hours. The smart board tracking and the KPI board, which we generated that went along with this, a key performance indicator board, created an analyst awareness that we couldn't have paid people for that type of a result. The result that we got was that the analysts as a team became so heightened in terms of their awareness that their productivity increased by almost 50%, and the quality improvements saved 950 full-time equivalent hours on the investigations that were performed and that were being formed as a result of a reduction in the deviations. And here on the right hand side you can see major and minor deviations that were occurring in the lab, and they had about 18 deviations which reduced to 10. That was quite a significant increase, and that's been driven down to such an extent that I think in 19, at least for the first 7 months, they didn't have one deviation in that laboratory, which was really quite remarkable. And all this was as a result of the types of lean implementation with the U-shaped configuration setup, and then also these notification boards. So what I'm showing you here is a spaghetti diagram. The left-hand chart, the right-hand chart is the one you've seen before in color. The left-hand chart kind of shows you what the laboratory footprint was when we started. And here on the bottom left is where the material would be logged in. It was logged in manually. It would be moved over here to a rack system. An analyst would come in, they would take this, they would do workups over here, maybe in a hood, they would do something here, it would come back, they would go down here, pick up glassware. You can see it's all over the place. By going to a U-shaped configuration, we reduced the deviations went down by over 60%. Well, actually, we're up to about 75% now with some facilities. And the cycle times in terms of getting the process out was reduced. At the time I put this together, it was 16%. Now it's up around 30%. So that's really quite significant, and it shows the real value of this approach. Just got one or two more. So the reduction in movement of samples speeds up operation. The reduction in the travel time is important. Errors are reduced, and the smooth flow improves performance. The reduced volatility means resources are more fully utilized, and the sample analysis increases. We almost get rid of backlogs. That was a big issue with this particular company when we first started. They were a contract manufacturer, and they were being hounded by one of their big international clients. After putting all this in place, they became their biggest champion of our client. They were very, very happy with the fact that we had got them to this level of performance. I mentioned barcoding and using tablets. This is a commercial drawing, but it actually makes the point. This is a industrial tablet that can be wiped down. It can be used in a clinical operation. You can see there's a vial here. This could just be the same with what we're using as a vial, not for a clinical application, but a vial for HPLC vials, for example, if the material is made up, that we have barcoded. The machine has a barcode, a scanner here in the end, and then it also has a tool here to allow you to write on the screen. So that's something that we've used as well. And then what we have here is, this is an analytical worksheet in this particular case, a lab notebook. And this is a water system using a similar type of a similar type of tablet to the one I've shown you, and this is their equivalent type of printout. The nice part about this is the primary data now. It's locked down, and this can be exported off to the LIMS system without any manual intervention or operator intervention, which is a nice feature because it reduces the possibility of data integrity issues, which is a big problem today. So that's just about an hour. I think I will stop there. I hope this has been helpful. I look forward to your questions, and thank you for the opportunity to share some of these ideas with you.
Thank you very much, Nigel. It was a very interesting topic, and the guys were texting me on my mobile saying that some of these tools can be very beneficiary and we can use it in the lab. Actually, please, guys, everybody, if you have questions, please write it down on the questions and answer sections. I will start taking questions from now on. There's a couple of guys who said they need the presentation material. We have to consult with our partner at Smart if they can provide the material or not. But you can contact Maysa. She just wrote her email and her contact number on the chat box. Please take her email and her contact number, contact her, and if possible, she can provide.
We're very happy to work with Aegon and Yara. As I'm sure she's explained, we have partnered with you now for 3 years, and yes, the goal is to provide partnered expertise. So if there are clients or client prospects or people on the call today that require help with this, then please contact Yara, and we will do our best together to help you with any aspect of this. Yes.
And if anybody of you guys needs further collaboration, further information about the work of Smart and Mr. Nigel, you can contact us. We can provide you with full information. Now I have actually 2 questions, Nigel. One of them is if the guy can or the lady can get the PowerPoint presentation, you have to consult with Maysa, please, Ru'ya. And the other one is from Imad. Imad says, can we consider the below key points to contribute to improve lean lab works and lab efficiency? First of all, implementing PAT approach, online devices, FTNIR, Raman, etc. 2, reducing samples, testing level and frequency of raw materials based on risk assessment of the supplier's qualification levels. I mean approved and authorized certified suppliers. The same approach can be applied to finished product analysis based on risk assessment and product quality review outcomes. 3, increase lean lab efficiency by maximizing the product manufacturing campaign as much as GMP compliance allow. So the waste time and lab chemicals and consumables quantity can be reduced.
Yeah. Yes. I'll answer the third question first.
Yes.
That was the point that I made in the first 5 minutes. Although we're considering the laboratory here and doing things that can influence the efficiency and compliance of the laboratory, one of the things that you learn in our business is that the solutions or some of the root cause problems for deficiencies in a particular area are not necessarily in the area that you're trying to fix. Some of the solution features are often related in another area, and the questioner is correct. And I made this point very early on. If you just focus on the laboratory, you may end up with a fabulously efficient laboratory that is very efficient, very compliant, which is great, But in terms of what is the value of that to the company and the factory that is making the product, if the product now just sits around at the end of the day, or it's made and it's QC'd and quality assurance, it takes 6 months before they release a product, then you've not really added. I think if you can, as I tried to show with one of the later slides, if you can coordinate the activities with the QA department for the release of the product, actually, as you complete the analyses, the individual test methods, then you provide that efficiency, that reduction in the total cycle time. Conversely, if you can also, and this is the reason why I showed you the smartboard idea, is if you can maximize the if you can minimize the turnaround time from manufacturing sending an in-process sample to the lab and getting it analyzed and getting that result back to them so that they can manufacture and continue to manufacture, then that's the real benefit. You will maximize the total cycle time of the plant in terms of getting materials in, making your product, testing it, and releasing it. And that is very, very important. I spend a lot of time, even with the client that I shown you where we put the board, they still haven't fully taken that on board. One of the things that's my dream, if you like, is to try and emulate almost single piece manufacturing, so that the raw materials come in and it goes off a production line, it goes off a filling line, and it could be all done in one step. The practical fact of that, our industry, because it's so highly regulated, it's very, very difficult to actually get to that ideal point. We're not building motor cars or electronic components. We're making medical products that are going to go into people, and so there are regulatory requirements that need to be met, testing requirements, stability requirements. We need to know, and so that makes it difficult. But what we need to do is to approximate to as near to those scenarios as we can. I think the second part of that question, the second question was, can you apply this type of philosophy to products, raw materials, and everything? And the answer to that is yes. You can even apply the same type of philosophy in a clinical lab environment where you're testing bioanalytical samples, and one or two client companies, and also one or two of the larger testing companies here in the United States do operate these types of philosophies to actually minimize cycle times. So yes, there is no limitation on which type of sample you can analyze and use this type of approach. Depending upon what type of analysis you're doing, it's going to be a little bit different looking at a Raman situation or an NMR situation compared to HPLC, things that I call rapid analysis. And you're not going to have 10 NMRs in a row. You're going to have one, but you can attack the analysis situation in the same way in terms of setting up how the samples come in for analysis. Once you understand what the uptime on the machine is, this is why it's important to do the value stream map, so that you actually understand. Remember that inch wide and a mile deep comment. You need to really fully understand the consequences and the constraints that you have for the particular type of analysis that you're trying to perform. Because once you understand that, then you can design a better future state. You can say, okay, we can't do this and this, but we can do something else. And well, what does something else look like? Well, something else looks like, and then you build the parameters around that. And then you set up, well, okay, if we're going to do this, we're going to make those changes. We're going to perform a Kaizen. We're going to do that exercise, that plan, do, act, check thing. And before we can check it, we need to set up, well, how are we going to measure if we've made an improvement or whether or not we've made things worse? Because sometimes you make a change and it makes things worse. I mean, you have to get realistic about this. The important thing is that you've made a change and you've measured it. And then if it's good, you might have only made a 10% change, and that's not good enough. And so you'll say, okay, well, if we change this same parameter again, maybe we can get a bigger payback. And so you'll go through the exercise again. And this is what I'm saying. It's continuous process. It's not, you don't get healthy by going to the gymnasium once. You go to the gymnasium several times a week, and you have a regime that you go through that maintains your health. I see this as manufacturing and operational corporate health. If you do the right things continuously, then you will get the benefits. If you only do it once, you will get a short-term payoff. But the likelihood is that within a short space of time, just like you would with your own health, if you don't keep up with the program, then within a short space of time, you will be back to square one or even further back than that. I can't remember what the first part of the question was, Yara.
The last part of the question was the reducing samples and testing level and frequency of raw material based on a risk assessment, taking into consideration the vendor approval procedure that they have. It's an authorized, approved vendor, so I can have a risk assessment and reduce the number of samples, and that would contribute to the lean at my laboratory.
Yeah, I mean, that's going to come down to if you're looking at material that's coming from a manufacturer that you're using in your process, and that as part of your vendor certification process, you're going to test that material, and you're going to test that material, each lot maybe that comes in, and you're going to have a scorecard, you're going to generate a metric, you're going to generate some data that says, okay, for lot number one, it was perfect. For lot number two, it was perfect. For lot number three, it was good. If you, once you have generated enough history, a database that shows the operating history of what you have, then you can usually request a reduced or come up with a protocol for a reduced testing, because you can show that, okay, we've tested, just to use an example, we've tested 20 or 30 or 50 batches of material coming from a particular place, and it's always the same because the quality is good, they must have good systems at their end, and the material is bang on. And so you can come up with a reduced protocol that says, okay, based on these results, we'll test once every 5 batches. The issue is you also have to have a default mechanism there. And the default mechanism is, as long as it maintains quality, we can test every 5 batches. But if the quality falls off, then you have to have a mechanism to revert to testing every batch again until you arrive at a situation where every batch is being tested and it's identical again. So it's like if you fall off the perch, then the consequences have to be a little bit severe, because there's a trust that's been broken between you and the you're asking a regulatory body to trust the fact that you've got your eyes on the ball with your supplier, and the supplier is not doing something. It doesn't mean that you're being lazy, that you don't want to test materials. You've made the case to your regulatory authorities in saying, look, we don't want to test as many because it costs a lot of money to do that, and we don't need to. It's a waste of time. they will accept that. But if it falls off the standard, then there has to be kind of a punitive requirement in the protocol that says if I fall off my perch, then for the next 10 batches we go back to testing every one of them.
Yeah, I would love to add to that for Imad. It's typically the switching rule in the military standard sampling plans. So in the military standard sampling plans, you have a switching rule which says that after a certain number of batches that passes, you can reduce your sampling procedure, you can reduce your testing procedure. But as Mr. Nigel said, whenever you go off the wagon and whenever you are out of specification, you have to go to normal testing. That means each and every lot, each and every sample. And maybe if you have continuous incidents of failures coming in different lots, that will take you to the tightened sampling procedure. So it's if you're doing the reduced one, you should be able to implement the tightened one as well inside your system. So this is for Mr. Imad's question. We have the last question from Ahmed. No, okay. Imad saying many thanks, Dr. Nigel and Diara. We are aligned with your input. Ahmed has another question. It says, is it possible to implant a small QC lab within the production site?
Yes, and some companies do that, particularly for in-process analysis, where again you want rapid turnaround, and it could be a single simple HPLC. You might have one or two HPLCs set up in an area, and you're looking for a particular intermediate, for example, a peak off an HPLC. And so, yes, there are, I can think of one factory that's not very far away from our office that is a big client that makes narcotics. They have a very substantial product testing lab that's in one part of the manufacturing factory, which again is very much like I described. It's stuck at one end of the factory, and then they have a small lab which is for in-process samples, which is right in the heart of the factory, and that is a chaotic place. I mean, it really is. It was that particular laboratory that we spent a lot of time working on some years ago to actually define some of they had a very, very peculiar login type of situation. They were working with paper analytical worksheets, and they got many situations where they were making a narcotic cake, and they had to get samples come from their dryers. They were drying cake, and they had to get dryer samples. They had to dry the material down to 90%. And so they would run their dryers, their spray dryers, and then they would, these drum dryers, should I say not spray dryers, these drum dryers. And every so often they had to take a sample to test for moisture. And every time a sample came in, because this material, once it was dry, it had to be put back into the chemical reaction within an hour or two, otherwise it would be spoiled. And you couldn't over dry the material. So they were taking a sample maybe every couple of hours to make sure that it wasn't too wet and it wasn't too dry. And so in the middle of a shift, you'd have these samples arrive. And as I used to say, it was like a bomb going off in the lab. It was absolute. As soon as the samples arrived from the dryers, it was like everybody dropped everything, literally, and worked on getting those samples turned around, because otherwise they'd lose the material from the dryers. It was and it created a lot of errors, and so we had to reorganize that, and that's what came out of the one of the smart solutions that came out of that was to go and move to scanning the samples in and moving to electronic worksheets. But also, instead of drying and bringing the sample in, I mentioned about don't look for the solution to your problem always in the place where you have the problem. Sometimes if you look at the adjacent space that is supplying material to your area, that could be the problem. And so one of the things that was a problem in this example, which I'll use as to illustrate the point, is you had a dry cake coming from the manufacturing dryers, and we wanted to know, it was putting the problem in the laboratory that, well, is the cake dry or not? So it was like everybody was focusing on how can we turn the samples around? And it was a case of the solution that we came up with was we're not even going to test the samples but once in the laboratory, and not 5 and 6 times now. What we ended up doing was we ended up putting a near infrared probe into the evaporation stream of the dryer, and as the material dried, a little bit like a tumble dryer with your laundry at home, as the airstream became drier and drier and drier, we set a set point there. Once it got to about 90%, it would kick off. And at that point, then we would take the material out and do a proper an analytical test of dryness in the laboratory. And what that did was it shifted 90% or maybe 95% of the analysis that was previously being done was moved to an electronic aid on the manufacturing equipment. And so this is an example of, again, thinking outside the box. Don't always think that the solution to your problem is in the space where you're trying to create the solution. Sometimes by looking outside, in this case of the laboratory, we were able to enact a solution that made most of the problem for the laboratory go away. I hope kind of that's practical.
Yeah, it's very practical and very detailed answer. Thank you very much, Dr. Nigel. I think this is all the questions that we have for tonight. I would like to thank you very much for your time, for your participation, for this very informative webinar. Thank you very much, dear. I would like to thank everybody who attended here tonight. I hope to see you soon again in our next webinar. Please stay tuned to the next webinars, and if you need any further information from us or from Dr. Nigel, please call Maysa. Her contact is still there, her phone number, or contact us on our social media. A recording of this webinar will be uploaded in our social media channels, as long as our YouTube channel. So if you need to go back there again to review some data, to share it with your colleagues, please feel free to do that. Thank you very much, everyone. And thank you, Argon team. Thank you for all the logistics, for all the help that you have shown today. Thank you very much. Thank you, everyone, and see you soon. Thank you. Thank you very much.
Bye.